So on to the next talk. It's me. Hi everybody. So what I'm going to talk about now, so we mentioned earlier that at least in the way that we have been analyzing the data for the last few years in Zurich and also some others, we depend on prior knowledge, some prior knowledge in the form of spectral libraries in order to analyze the DIA or SWATH data. And this usually means some kind of DDA, classical DDA-based characterization of the samples that we're interested in. So the goal here is to kind of bring you up to speed on how to generate these spectral libraries, at least the way that we do it. And this will also be the topic of the first tutorial that we're going to do directly after this. So pay attention, and then you can do well in the tutorial. So why do we care about prior knowledge in DIA analysis? I think this has probably already been covered, but I'm just going to quickly repeat this in case it's not clear. And then I'm just going to try to walk down through this procedure and highlight what each of the steps are, step by step, and point out a couple of issues that we think are important in this procedure. And then also Tina mentioned that there's a lot of publicly available resources online now, peptides and proteins that have already been characterized by various different groups, and I just make a few points about this, say what these are and whether you can use them or not. So first, why do we care about prior knowledge? So this, I already showed this to you, right? So this is the problem statement. We have these three precursors that are in the same isolation window. We fragment them, and we get all these fragment ions in this multiplexed MS2 spectrum, and we can't interpret it in the classical way. So I also showed you this already. This is our SWATH MS2 spectrum and the peptide that we're trying to dig out of there. And so if we have a reference spectrum for this peptide, then we can go looking specifically in this spectrum for the signals that correspond to this peptide, and we can do extracted ion chromatograms of these fragment ions, and now we get clean data out of this kind of mess of fragment ions here. So of course this requires this, right? So some years back when we started presenting this type of method at meetings, we would say, okay, if you have some DA data and combined with some prior knowledge in the form of a spectral library, and you put these things together and do this sort of so-called peptide-centric data analysis, then you can come out with nice, quantitative, robust data. And then people would say, but where do you get these spectral libraries from? So this was a kind of source of confusion for some time, and I think people who are in the targeted proteomics world knew something about this, but others not so much. So what we decided to do was to write this down in kind of protocol format a couple of years ago in this Nature protocol paper. And so what I'm going to present to you now is basically the contents of this protocol paper, and also what you're going to do in the tutorial is based strongly on this paper. So the goals here were to kind of document what we do for library generation in this lab, but also to kind of document and point out some kind of best practice issues, at least from our perspective, and also to document how libraries that we might make available from some projects were made, so that it's transparent if people want to actually use these, how they were made. And so this table you've seen before. So this is the goal, right? This is what we want to get out. We want to have a table where we have the peptides that we're interested in. We know what proteins they come from, of course, and we know some mass spectrometric information about them, right? So what are the precursor and fragment ion masses? What are the charge states of the precursor and fragment ion? Which fragment ions are the most intense, and what are the relative fragment ion intensities for those fragments? And also this peptide, the retention time in this normalized space, and I'll say in a few minutes what this normalized space means. So that's why we care. So now let's look at the procedure. So those of you who are familiar with DDA data analysis will recognize much of this workflow. So we're doing some typically DDA data acquisition on some samples of interest. We need to convert these data files into file formats that we can manage, and we need to do peak picking, right? So the search engines need a peak list so you have to do some centroiding and peak picking. Then we do a classical kind of database search using Sequest or any of the other tools. We do some FDR control of these database search results. And so this part of it, probably many people who do DDA will be very familiar with. The next thing that we do is to combine the database search results into a spectral library. And so this is mainly to reduce redundancy. So if we have a particular peptide that we identify multiple times, we only need one representation of this spectrum in the library. And so this is so-called spectral library generation. And then we need to take this spectral library and we need to apply a set of filters to decide from these spectra what are the fragmentines that we actually want to monitor in or to extract in this peptide-centric analysis. And so this is the workflow. This is the kind of simple version, but if you add in all the details, it looks basically like this. So this is now rather complicated looking, but it's just a way of saying the same thing with the details added, right? So we do data acquisition, we do centroiding and file conversion, either using this ProteoWizard MS Convert tool, or we also have an internal tool called QTOF Peak Picker. We do database searching, typically using Xtandem or Comet or some other search engines. We do we sort of post process these database search results using Peptide Profit to basically essentially estimate the error rates and also to put the database search results from multiple different search engines into a common framework, which is the Peptide Profit probability, and we can then combine the search results from those various search engines together with a tool called iProfit, and then this increases the power over a single search engine if we combine multiple of these. Then we do some FDR control, including at the protein level, using a tool called Mayu. So these tools here are all part of the Trans-Proteomic Pipeline, which is a set of tools out of the Institute for Systems Biology in Seattle. And we then also is Spectra. So we then move on to do the spectral library stage, where we combine the spectra. We reduce the redundancy of spectra, where if we have multiple instances of spectra for a single peptide, we need some way to get that down to one. At this stage, we're also doing this retention time normalization, so I'll mention the details of this. And then once you have the spectral library, it's just a matter of filtering this out, as I said, to get a set of parameters that you can then use in one of these downstream softwares. Like we're going to use Skyline, and we're going to use OpenSWATH during the week. This is Spectronaut, which Lucas Reiter will talk about probably on Friday. PeakView is the one from SciEx, and by now I'm sure there are others. Okay. It's also important to mention that this is our workflow, and you may be forgiven for saying that it's rather complicated. It is, it's kind of multi-step, but having a multi-step workflow like this allows you to kind of control the parameters at each stage. But there's also a more simple way to do this using Skyline, right? So you can use whatever your favorite search engine is here, and Skyline will also handle all of these parts, the spectral library part and the filtering to get this actual set of peptide query parameters. So I think Brandon will say something about this, but it's important to mention that there are alternatives. And so the first question to start with is, what kind of samples do you want to put into this analysis? So you could imagine many different things, right? The most simple and kind of default choice here is just the samples of interest, right? So this means the same samples that you're going to analyze using a DIA method, you can analyze those by DDA and generate a spectral library from this. The next one is to take these same samples of interest and do fractionation at the peptide or protein level, and this allows you to get a little bit deeper and more comprehensive library than you would from just this kind of single-shot analysis of the samples of interest. And this might be worth doing because, at least in our experience, we find that the single-shot DDA analysis is less sensitive in some cases than the single-shot DIA analysis. So that means that in order to exploit the DIA data most completely, you would need kind of a more comprehensive or deeper DDA analysis of the samples of interest in order to reach this. So this is why we would typically do some kind of fractionation. There are also many options, though. People have tried to synthesize peptides using this crude peptide synthesis that what Tina mentioned. So this you can do relatively cheaply for sort of 10 or 20 bucks per peptide, and you can synthesize these at large scale. And people have also done these at very large scale, organism-wide scale, and some of these resources are available. One problem with this is that you have to know in advance what the best performing peptides are, and as Tina mentioned, this is not necessarily straightforward. It's difficult to predict computationally. And so another alternative is to use full-length recombinant proteins or synthetic proteins. And if you can get your hands on these, then this is a nice option because it tells you, the information falls out about what the best peptides are immediately. You see, you analyze all the peptides for the protein essentially, and this tells you which ones digest best, which ones have the best response factor in the mass spectrometer, and so on. And there are now efforts to do this on sort of medium to large scale also. And then there's the idea of kind of public or organism scale libraries, which can be kind of a combination of any of these things. Either you synthesize all the peptides you can think of, or you express all the proteins you can think of, or you analyze take all the DDA samples that you've ever analyzed and analyze them all together and combine everything into one big giant spectral library. And these are also, this has also been done, and it's also a possibility to use these. So these are the choices. But in the end, the output is still going to be something like this, which is a table that tells you how the peptide looks to the mass spectrometer, and where you should look for it, essentially the coordinates. Okay, so one thing to point out. So I mean, we're going down the workflow here. So sample choice actually, I guess, is up here somewhere. I didn't write it, but now we're on to DDA data acquisition, and I will just try to make a few points about each of these topics as we go down. So in terms of DDA data acquisition, there's maybe one thing to point out that it's not completely obvious, is that there may be some benefit to trying to increasing the spectral quality over what you might have for a standard DDA analysis. Because one thing that we want to have is to know is the relative intensities of these different fragment ions. And a higher kind of signal-to-noise spectrum will give you a better representation of these relative fragment ion intensities. So sometimes what we do, especially if we're doing upfront fractionation, we can increase, for example, on the triple top, this would be to increase the scan time up to 100 or 150 milliseconds, and the normal scan time that's recommended in DDA mode will be 50 milliseconds, but increasing the scan time gives you better signal to noise in the spectrum, and so this may have some value. You can also do things like setting up the dynamic exclusion settings in your DDA method such that you get more than one MS2 spectrum per precursor. For example, by reducing the dynamic exclusion to half of the chromatographic peak width, meaning you might get a DDA event, a MS2 scan event triggered at the apex of the chromatographic elution, and thereby getting a higher signal-to-noise spectrum as well. And so this can be beneficial. And you may also want to consider the collision energies, because in DDA mode or in SWATH mode we can't. So in DDA mode, when you try to collect an MS2 spectrum for a peptide, the machine computes the appropriate collision energy, right? And that's based on the charge state, and it's based on the mass charge ratio. But in DDA mode, because we just have this large precursor isolation window, there could be several different peptides in there which have several different charge states. So we can't apply a specific collision energy that's really specific to any one peptide. So what we have to do is kind of say, okay, we'll compute what we usually do is we'll say we'll compute the collision energy for a +2 precursor and apply that collision energy. And if you want the spectral library to match the DIA data in terms of these relative fragment ion intensities, you can, if you wish, collect the DDA data using these DIA collision energy settings. So meaning that if you see a +3 peptide, you pretend it's a +2 peptide, and it may get over-fragmented, but at least the fidelity between the sort of relative fragment ion intensities that you see in DDA will be maintained with those you see in DIA. So these three things is not something that we've tested necessarily very systematically, but have kind of tried to well, essentially what I want to point out is that these things might reduce the number of total identifications that you get from the DDA. Yeah. But yeah.
So in DDA and DIA collision energy, so do you use high collision energy in DIA with respect to DDA, or is lower than DDA?
Well, so
how do you decide that?
We use so I mean there's something known about what the appropriate collision energy is for various peptides in a given charge state, right? There's some equation that accounts for the mass and for the charge state which should give you some kind of optimum fragmentation. and in DDA, you know the charge state, right? So the machine says, here's a +2 peptide. I now run.
So for example, if you use DDA, let's say a value of 27, that's what we use on Q-exactive generally for +2, +3 charge peptides.
Yeah.
So in that situation, how we're going to decide energy for DIA?
So what we're going to do is, or what we do is to say, we're going to assume everything is +2. We're going to calculate the collision energies for +2, and we're going to take the mass to put into this equation from what we know is the precursor isolation window, right? So this will be now 10 or 20 mass units wide, but we know it's somewhere in that range, right? So let's say we take the middle of the window and we assume it's a +2 peptide, and we use that to calculate the collision energy. So we know that this is not going to be appropriate for all of the peptides that are in the window. In particular, the higher charge state peptides will probably be over-fragmented. What you can do, at least on the SciEx platforms, I'm not sure about the thermal platform, but what you can do is a ramped collision energy, right? So you can say, I'm going to have a particular voltage, but I'm going to start at -5 volts, and I'm going to ramp across to +5 volts over the course of the scan. And this sort of somehow gives you some buffer that might allow you to account for this, right? But my point is that there may be some value to sacrificing some identifications in order to get spectral quality and spectra that will represent well what you see in the DIA analysis. And this is particularly true, I think, if you're doing fractionation experiments where, yeah.
Just one question to this. Isn't then, what is an advantage then to reduce the dynamic exclusion over just running more replicates to construct the library?
Well, you can do either. I mean, the advantage is that you get more MS-MS spectra. And in particular, so many people would set the dynamic exclusion to full chromatographic peak width, right? And that means that you would get, so the peptide starts to elute, and then you get an MS-MS spectrum when it's on the way up. But you don't necessarily get an MS-MS spectrum at the apex of the chromatographic elution. And you can imagine that if you took the MS-MS spectrum at the apex of the chromatographic elution, the signal is going to be higher, and you might get a more high-quality spectrum. So by setting this dynamic exclusion to like 1/3 or half of the chromatographic peak width, then you have a better chance of triggering an MS2 spectrum at the apex, and therefore getting a higher quality spectrum. But this comes at the cost of not, choosing to collect an MS2 spectrum on some other peptide, right? So you may get less IDs, but you will get higher spectral quality in the end, probably.
So then do you do it like this, that you run for like the same samples with different dynamic exclusions also to cover the other peptides?
You could do that. So this is not something we've tried, but...
Because then if you do the spectral library construction, then everything is averaged, right? Everything is kind of put together to get the best spectra.
So I'll come to that in a little while. There are choices to be made there. You can do this kind of averaging, or you can pick the best one. But if you're going to do multiple injections, I don't know whether it might benefit you more rather than fiddling with the dynamic exclusion settings, if you're committed to do multiple injections, then you could just do a fractionation of the sample and then inject those fractions, and then you would thereby get deeper coverage as well, and better spectral quality and so on. But so there's certainly ways you could imagine to optimize this, and we haven't necessarily explored all of those. Okay, so then we need to take the DDA data. It needs to be centroided and converted into a format that we can deal with in our pipeline. And one thing I just want to point out, there's several different converters available. Most people are using the ProteoWizard converters, I suspect. But at some point, we realized that depending on which centroiding algorithm you use, you can get a little bit different looking spectra. So this is three versions of the same MS-MS spectrum, but which has been centroided with 3 different algorithms, right? So this is a tool that was written in our group. This is MS-Convert using the ABSciX vendor libraries, and this is another ABSciX tool, ABSciX converter, using what they call protein pilot mode. And so this comes from exactly the same raw spectrum, but the relative peak intensities look rather different, right? And what we noticed is that, well, in fact, this tool was written specifically for this purpose, but if we now go and look at the SWATH data and look at the relative fragment ion intensities here, we can see that they're in best agreement with this tool. My understanding now, Brendan tells me that this was a bug actually in MS Convert, and they were taking the peak height under the mass spectrometric peak instead of the peak area, and this has now been fixed, I think. So this should probably look more like this one or be more comparable to this. But this is rather just to point out something to keep an eye on, right? If centroiding, peak picking tools that are optimized to get the best DDA search results might be doing some kind of weird processing that you don't know about, some charge state deconvolution or some fiddling of the ion intensities to improve the search results, but it may not necessarily be the best thing if what you actually want is to match it to the DIA data. Okay, so database searching, I guess most people know about. We tend to use these search engines, Xtandem and Comet. We use Peptide Profit and iProfit to post-process these results. I only mentioned that we very often are using like multiple search engines, usually a minimum of 2, but sometimes 3 or 4. And these are combined. If we analyze all of these independently with Peptide Profit, this puts them on a kind of equal scoring framework in terms of this peptide probability score, and that allows you to combine them in a kind of statistically sound way using this iProfit tool. And so what you see here is how many identifications you can get from a particular DDA data set using any of these search tools individually. But if you kind of mash them all together in the appropriate statistical framework, then this is improving the sensitivity, and we typically do this. So FDR control is another issue. So we don't. Well, so at this point, I think it's interesting to talk about what happens if you have a spectral library, but there are some errors in the spectral library. How will this affect this when you go to do peptide-centric analysis of DAA data? So let's imagine that there's a spectrum here. This is an MS2 spectrum, and for whatever reason, this is a bit lower quality spectrum. You get the wrong peptide sequence assigned in the DDA analysis. The search engine says this is some peptide sequence, but it's the wrong one. So what's going to happen when you use this spectrum to extract data from DIA SWATH? Well, what's going to happen is that if this is some different peptide and all these fragment ions come from a single peptide, but it's just not the one that you think it is, then you're going to extract all those fragment ions, and it's going to match beautifully with this library spectrum that you have. And so the error that you've made in the DDA analysis is going to be propagated here, and so the peptide sequence that you assigned here will also be propagated to here. And now this is bad. You've propagated. So this is not a good situation. You can also imagine a situation where the wrong peptide sequence was assigned in the DDA analysis because you have a chimeric spectrum, right? So this happens relatively often also in DDA analysis, that you co-isolate 2 precursors and you have a mixture of fragment ion signals, and this kind of confuses the search engine, and then you get the wrong peptide sequence assigned for this reason. And the question is, what happens when you then go to the DIA data? Well, what will probably happen there is that you will extract 2 signal groups, right? Because it's likely that these 2 peptides won't exactly co-elute. Or they might, but it's more likely that they won't. And then these will get scored badly, because one of the things that we said is important is that all the fragment ions should co-elute in this peak group, and if they don't, then the algorithms will score this very badly. So in this case, the error gets kind of resolved. Nevertheless, we don't really know what's the kind of prevalence of these types of problems, one or the other. And so I think the conservative thing to do is to be relatively strict when you're creating the DDA library, that you don't allow too many of these wrong peptide sequences into the wrong assignments, wrong peptide spectrum matches into this library generation. And so we use a software called Mayu to do the false discovery rate estimation, and this is happening both at the peptide spectrum match level, but also at the peptide level, at the protein levels. And the reason why this is important, so here's a graph of the estimated FDR at the peptide spectrum match level, the peptide level, or the protein level for a given data set, right? So at some FDR, and then on the x-axis is this iProfit score, which you can essentially think is like a database search engine score, right? So you can see at a given score, you can have a very low false discovery rate at the peptide spectrum match level, and a bit higher at the peptide level, and significantly higher at the protein level. And the reason why this happens is because if we look at this picture here, so in this depiction, these green guys are the target hits, and the red ones are the decoy hits. And so what you see, or the true positives and the false positives, And what you see is that, and these hexagons are the proteins, right? So these peptide spectrum matches, the ones that are correct tend to build up on the same proteins, but the ones that are not correct tend to be randomly distributed throughout the proteins, right? So this means that if you count the false discovery rate at the PSM level or the peptide level or the protein level, because of these kind of uneven distributions, the proteins being on the target proteins, the distribution they kind of build up, and on the false positives they're distributed all over the place. The problem is that you get false discovery rate accumulation at different levels. So this is just to say that it's good to know the false discovery rate at the PSM, at the peptide, and at the protein level, and then you can appropriately set the thresholds to decide what you're going to allow into your spectral library.
And Ben, just quickly,
yeah.
Maybe it's worth pointing out that that's just for 3 replicates,
right?
And that those peptide and PSM lines will diverge more if you were to do it over 100 replicates.
Yeah.
The more replicates you add, the further that red and blue line will get apart from each other. So it looks like there's a really extreme difference between protein and peptide FDR, and you should, but as you add more replicates, you're going to get a more extreme difference.
Mm-hmm. So this software Mayu actually that we used was designed specifically in mind to look at these large DDA data sets. Okay, so what about the spectral library stage? So there's a few things that are happening here. So now we have the search engine results, we have the FDR estimated, we've got some threshold where we've decided we're going to keep a certain fraction of the PSMs that we think are correct or correct at some false discovery rate. And we're now going to take those spectra and make a spectral library. So what I mentioned before is that typically what you have is many instances of a peptide spectrum match that map to a particular peptide, and you have lots of spectra for a single peptide. And the question is, what do you do with all these spectra? How do you choose whether you want to take the best one or combine them all together? Another thing we do at this stage is this IRT transformation. So I'm going to say what this is now. And there's basically some quality control steps here at the spectral level as well. So what about IRT? So this has been mentioned now several times, but not really the details. So the problem that's trying to be solved is that from run to run, or if you change columns, or if you go from instrument to instrument, the retention time of the peptides in absolute minutes terms will change. And so what we would like to be able to do is we would like to be able to normalize between one run and another. And in particular, in this case, we would like to be able to have the DDA spectra that we collect, the retention times for those, we would like to be able to project those onto the DIA data. So the way that we do this is to spike in a set of peptide standards. So these are 11 peptides which are spiked in to actually all of your samples, and they're kind of like nicely distributed across the whole gradient, and they're spiked in at a relatively high level, so they're fairly easy to detect. And what we do is we find what is the retention time for each of these 11 peptides, and we make a linear regression with the so-called IRT values for these 11 peptides. And these IRT values are just a set of it's an arbitrary scale, basically, that was defined when the method was developed, where the second peptide is 0, the last peptide is 100, and you just fit a line through the rest of them, right? And the reason why they use the second peptide as 0 instead of the first peptide is that this first peptide tends to be a bit more variable. So we're just going to say that this second peptide is 0, this last peptide is 100, and we're just going to draw a line through this, and we're going to project. So for our target peptides of interest, for A, B, and C that we detect in our DDA runs, we're just going to project the measured retention time through this linear regression onto this IRT scale. And then what we actually store in the library is this normalized peptide retention time on this kind of arbitrary IRT scale. And what this allows us to do is then when we go back to when we now go to the DIA data, we also have spiked in all of these peptides into the DIA data, so we can detect these and now notice actually on the y-axis here we have a 70-minute gradient, and now I'm putting a 35-minute gradient. So the gradient length doesn't matter too much because it's essentially a linear kind of compression expansion. And what we do is we look for these 11 peptides in the DIA analysis, and we say, okay, we look for these 11 peptides in the DIA analysis, and we redraw this linear regression, right? So this is the retention times of these 11 peptides in real retention, in absolute retention time now in our DIA data. And we can now go to our spectral library, our peptide query parameters, and we know what the normalized peptide retention time is. So we just plug this back into the linear regression now. So if this peptide C had a IRT value of 80, then we go back to the linear regression, and we can predict what the retention time exactly in this run should be, right? So we're trying to just basically transfer the previous knowledge from when we have an empirical observation of the retention time of this peptide, and we're trying to do it in a way that accounts for the slope of the gradient or differences in instrumentation and so on, differences in LC setup. And this allows us to make kind of fairly accurate predictions about where this peptide will appear in the new data. And so this is, and what this allows us to do is say, okay, I expect this peptide to appear at this particular retention time, and you can only look for it within, like, say, plus minus 5 minutes of where you expect it to be. And we know what the sort of accuracy of this is, and we can say with kind of relatively good confidence that the peptide should be there. So this actually improves selectivity by allowing you to not have to consider signals outside of this range, and it also can improve computational performance because you don't have to look at all of the data, you only have to look at this 10-minute window to see where the peptide is. Okay, so that's one part of the sort of this spectral library generation step. This Spectrast software tool that we're using is called Spectrast. And the second part is how do we remove these redundant spectra. So let's say we have 3 samples that we injected, and for this particular peptide APN, we have a DDA. The spectra, the MS2 spectra was triggered twice in this sample run, sample 1, and we have 2 spectra for this peptide here. In sample number 2, it wasn't there at all, but in sample number 3, it was only once, right? Because the intensity of the MS1 it might be different across these 3 runs, or it might just be for stochastic reasons, as we mentioned in DDA earlier. Sometimes you get it, and sometimes you don't. So now we have 3 spectra for this one peptide, but we only need 1, right? We only need 1 representation of this peptide. So the question is, what do we do with it? And this is basically where you have 2 choices. You can either say, so we now have a score, right? A database search score for each of these spectra. So we can say, oh, I'm just going to take the best one, right? This is called best replicate spectrum. And I think this is more, I think this is what Skyline is usually doing. Or you can say, we're going to take some consensus of these spectra, these different spectra that we have for this single peptide. So this is essentially an averaging step, and there's also some kind of QC going on at this step as well. So if there's one spectrum that looks completely different to all the others, it'll get kicked out and so on, and you have a kind of majority voting scheme here. So then what you get is this consensus spectrum, as you mentioned. And what we found in the past is the relative fragmentine intensities that we get from this consensus spectrum are a little bit better in terms of, so if we look at, compare this now to the DIA data, the relative fragmentine intensities we have there, we get a little bit better match, but so if we just take the best replicate spectrum, or we average over 5 spectra, or 10 or 15 going all the way up to 100 for a given peptide, we have a little bit better matching between when we do this consensus matching. Although it's only a little bit better, it's an optimization. So
I just wanted to interject quickly that we have since changed the way that Skyline chooses. We still use a best spectrum, so when you look in Skyline, you're actually seeing a physically measured spectrum. Okay. But we've changed the way that it's chosen from that, which was the original publication, and we haven't published the new method. But we found when we went to PRM that then PRM you get a spectrum like all the way across the peak, and what we found was that the best spectrum method that was probably used there was picking really noisy spectra because they happen to match to each other really well. So now we've changed to an algorithm that consistently in PRM picks the apex spectrum. Yeah. And I can't see any difference in how. But
if we're talking about DDA data though, so in DDA data, all you have to go on is this.
My guess is that it's through an algorithm that we now pick like the best scoring spectrum and then the most intense, and so there's a whole rationale to it. The original best spectrum algorithm was just to do an all versus all and pick the thing that matched everything as well as it could possibly match.
But so I think this was done in Spectrust, right? Even the best replicate, and I think in this contest, I think best replicate means best search engine score.
Okay.
No.
I don't know. Like I don't, because there's a published algorithm from BiblioSpec that I know that he has wanted to do comparisons with, but we've changed. We don't use that anymore. Anyway, I don't know. I haven't been seeing. I don't actually see real differences these days. But
in PRM data or in DDA
what?
data? In PRM data or in DDA data?
In DIA data. In terms of how Spectra's consensus spectra versus Skyline chosen real spectra perform for DIA data.
But what are you comparing to what? You're comparing DDA data to DIA data, or you're comparing DIA data to DIA data?
I'm comparing DIA results to libraries built with consensus spectra versus libraries built with.
But the libraries are being built with DDA data.
Yes. Yes.
Okay.
In both cases.
Okay. So that would be interesting to look at. I mean. Okay, so final step is then, so now we have a spectral library where each peptide only has one spectrum associated with it, either using a best replicate or a consensus approach. And we have these normalized retention times and so on. And now all we have to do is decide which fragment times we're going to monitor for the DIA analysis. And typically what we recommend, but of course this is open to some optimization as well, would be about 5 to 6 fragment times per precursor. Usually we have 6 actually. We select 1, 2, or 3 charge state fragment times. We go in this range 400 to 2000, which is related to the mass range of the instrument, but below 400 tends to be rather noisy. We select B and Y ions, assuming we're doing a label-free experiment and not doing what Tina mentioned earlier, where you might have to kick out the B ions if it was a Silac experiment, for example. We can include neutral losses, although these are not necessarily the most selective transitions. We generally exclude fragment times that are in the precursor isolation window for the reason that there tends to be a lot of unfragmented precursors there, and this tends to be a very noisy range of the MS2 spectrum. So for example, if your peptide is at like 515 and your window is from 500 to 525, then we select fragment times that are less than 500 or more than 525 and not in that range of from 500 to 525, because you tend to get a lot of noise in here. And for OpenSWATH, we convert to this TRAML, which is this PSI standard for file format for containing these types of libraries, and we add decoys, or else we just leave it in this TSV format, which can be used by these softwares, but in OpenSWATH we normally convert this to TRAML. And so that's the end. So then now we have this result, right? So this is the table I keep showing you, and we have it either in this TSV format or the TRAML format, and so now we're ready to import this into the software that's going to do the analysis. So summary is, okay, we just walk down through this, choose what kind of samples you want to use. So either representative samples from your project, or synthetic peptides or proteins, or potentially a mixture of these, or potentially online resources that are large collections of these. The spectral library relative fragment ion intensities, it's good when these agree with the SWATH data, so meaning that the relative, meaning that when you don't allow peak pickers to change the intensities too much. If you have better spectral quality, then this will agree better with the SWATH data. Well, we just had a discussion about this. Our experience has been better for consensus in the past, but Brandon says maybe best replicate is also potentially an option. Control the false discovery rate of the DDA data, and it's worth looking at this at the PSM, peptide, and at the protein level. And it's worth transforming these retention times into this normalized space, and then you can get very accurate predictions of where the peptide should be when you see it in a new run. And so this is all covered in a lot of detail in this paper I mentioned, so you can take a look at this if you're interested. Oops, so now I'm over time. I'm just going to go quickly through this publicly available spectral library resources. So we realized at some time that we were doing, okay, here's SWATH project 1. Now we need to do analysis number 1. Then we need to develop an assay or a spectral library for this set of samples that are specific to this project. And then we're doing more projects. And then we said, oh, maybe we should sort of pool these together and make one big library. And so this is what we started to do. So in the case of human samples that we were analyzing in the lab, we basically took everything that we had, which is a whole big mixture of cell lines and tissues and plasma and affinity purifications and all sorts of stuff, kind of with fractionation or not. And it was just a whole big mess. And we searched it all together using this exactly the workflow I've just described. And we came up with a data set that had about 10,000 proteins in there, identified from about 140,000 peptides, which translated to almost 200,000 precursors. And we had six fragment ions for each of these precursors. So this is more than 1 million fragment ions. And so that's what is depicted as CAL here. It's called Combined Assay Library, although colloquially people call this Pan-Human Library. And what's being shown here is the blue is the library that we made. The green is Swiss Prot, this 16 or 15,000 that Rudy mentioned earlier that have good protein level evidence. And the pink is the missing stuff, right? So we basically are overlapping with the things that have good evidence as have been detected before, but we don't cover the whole thing. And just to show that this kind of works, we took this large library and we took another data set that had been, or SWATH data set that had been generated in a different lab. This is the lab of Anne-Claude Gingras. And what she was interested in analyzing was APMS experiments, so pull downs. And there was about 150 proteins of interest that they were interested in the study. So what we did was pull out all of the coordinates for all of the peptides from these 150 proteins out of our large library, and we analyzed her SWATH data with our library. And the results are essentially shown here. So, of course, we had a different set of peptides than she had in her library because she had generated these locally using DDA analysis. But the results were more or less in good agreement, at least in terms of the trends. So what you see here is that the pink and the green is the fold change for the two conditions analyzed using her local library, and the blue and the purple is the fold changes for these proteins analyzed using our library. So there are some differences, but from a biological perspective, you essentially get the same answer, and this was sort of a nice indication that what we were doing in terms of transporting libraries from one place to the other was not completely crazy. And so now you can see this is a portal from the Moritz lab at the ISP Seattle called SWATH Atlas, where they're hosting some of these libraries, and you can see that there's various species here and what their coverage are. So this is this human library, which has about 50% coverage. Tuberculosis is a sample we worked a lot in our lab, and so this is a very comprehensive library for that, which includes both synthetic peptides and kind of real samples, real bacterial samples measured, and various other species. So if you're interested, you could take a look at this. And we have all these various different formats available there, and you can potentially try these if you want. Since this has happened, though, there's been a lot of developments in terms of people trying to do this on a very large scale or do this in various ways. So one is this SRM Atlas, also from the Moritz lab, where they synthesized I guess it's 158,000 peptides for human, which covers 99.7% of the human proteome, and they measured these mostly on QTOF, I think, and then they deposit the data in this repository. The one problem with this is one that we mentioned earlier, is that I think a lot of these peptides were chosen based on computational prediction algorithms of what the best peptide might be, and it's turned out that these don't work particularly well. So it means that some of these peptides will be good, but some of them may not be so particularly useful. There's another resource now, a paper just came out from Bernard Kuster's lab where they're trying to do something similar, and I think the peptides from this project are somehow maybe a subset of the peptides from this project, that they combine them together, at least some of them. And what they've now synthesized is like 330,000 tryptic peptides, and they've measured them in a similar way to here, this crude synthesis method, and they've measured them on a fusion, either in the Orbitrap or the ion trap, measuring the fragment ions, and they use different activation modes, and like HCD or CID or ETH HCD, and all various different combinations of activation methods that are available on Illumos, and also different activation energies. So there's a very kind of comprehensive set of spectra that represent different ways of fragmenting the peptide, at least on this instrument. And they now have sort of 330,000 of these, but they're which covers again the whole human predicted genome, but they're also planning to include things like PTM peptides and sequence variants and non-tripic peptides and MHC peptides and all sorts of cool stuff. So this is now available too, and could, although we haven't tried this could potentially be used for this purpose. Another one that's nice, I think, is from the McCoss lab. I guess Mike will mention this later in the week, where they are now, instead of doing this synthetic peptide approach, are making full-length proteins using in vitro transcription translation systems. And this is a smaller resource at the moment, but it's more information-rich, I would say, in terms of this is a graph for one protein, right? And you can see, and these are all the different peptides that are observable from this single protein, and you can see which peptides perform really well, and you have nice empirical information about this, and so that's quite attractive, and I guess Mike will mention this later. So one question that comes up sometimes is, what about moving between instruments? So is the fragmentation similar if you go from one instrument to the other? And the sort of short answer to this is to say instruments that use beam type CID or referred to as like HCD on the Orbitrap spectrum, if you compare those, then they look similar. So in this graph, what I'm showing is always comparing the relative fragment ion intensities of a large number of spectra to SWATH MS data collected on the triple TOF, right? And on the left side is the spectral library, which comes from several different sources, either Orbitrap Elite HCD, Orbitrap Elite CID, again, Orbitrap XL CID, or triple TOF shotgun data. So you can see that the same, coming from the same instrument, has the best match, but also the HCD data from the Orbitrap looks pretty good, whereas if you go to CID data from an Orbitrap, it looks much less good. So I mean by CID, resonant excitation, like ion trap cell fragmentation. So it seems like it's good to keep the libraries on the same instrument, but if you switch between instruments that have kind of similar fragmentation modes, then this also seems to work pretty well. This is also a spectrum comparison from this, also from this Bernard Kuster resource, and here they compare QTOF spectra from our lab to beam type CID or HCD spectra from an Orbitrap, and they say, okay, they're pretty well correlated, and most of them are pretty well correlated. So it seems that there should be some transferability between these, and these large-scale resources to some degree are reusable between labs and between instruments. Same thing for retention time. This is now a study I mentioned when talking to Edward earlier, where we collected SWATH data from a lot of different instruments in a lot of different labs. And this is the data. So what I'm plotting here is the deviation from the predicted retention time using this IRT method, right? So we're usually within a couple of 100 seconds of the predicted retention time. And this is the data from here, from Zurich, using one particular chromatographic setup. And so we're using our library to analyze our SWATH data, but now there's other sites using their library to analyze using our library to analyze their SWATH data. There's actually some small differences in the chromatographic setups. So what you see is that the errors get a bit larger here, and this one gets, again, a bit larger. Here's the standard deviations, but it's still definitely useful information. So it's still useful, but I mean, the caveats here that if you make dramatic changes to the chromatography system, like changing the mobile phase modifier or heating the column or something like this, then these spreads will get larger again. So I think we're out of time, so we can skip the summary. I mean, I guess I just would say in terms of outlook, this is our workflow where we have prior knowledge based on a spectral library based on DDA data, and we do peptide queries in the SWATH data. So we generate peptide query parameters, and we analyze the SWATH data doing this. But there's, of course, now going to be other methods to do this, which is what you're going to hear about later in the week. So, for example, this DIA umpire tool, which allows you to generate the spectral library internally from the DIA data. So this doesn't require any DDA data, but you just say, so this will be explained to you in a lot of detail. Or, for example, from the McCoss lab, there's a tool called Pecan, which is not requiring spectral prior knowledge in the form of spectral libraries, but rather just attempts to directly interrogate the DIA data using in silico generated peptide queries, essentially. So you'll hear much more about this. I think it's interesting to note that these two approaches, which don't require the prior knowledge, I think there's some evidence that they work better when you increase the quality of the data. So as the instruments get better and the windows get smaller, the selectivity gets better, these work better and are more comparable with the performance of this. But this is something that will come up, I think. Okay, I'm sorry I went over time. Is there any questions right now? If not, thanks for your attention. Yep.
Hi, so my question is, can you use your workflow to create a library if instead of standard retention time peptides you are using endogenous ones?
Yes. The short answer is yes, I think. So these spiked-in peptides are really just a convenience, right, to make sure that you have something that's always in every sample that's the same. There was a visiting postdoc in our lab called Sarah Parker who came with a bunch of data, and she wanted to analyze the data, and she didn't have all these IRT peptides spiked in. So what she did was looked across lots of data sets to find out what were the most commonly identified peptides in terms of like, keratins or trypsin or filamentous proteins that are well conserved across species. And she came up with lists of peptides that tend to appear in most kinds of samples, at least in like yeast and human and mouse she looked at. And even in phospho-enriched peptides, you still see most of these actually. And she made lists of these and checked whether they work for this purpose, and they seem to basically work most of the time. So she has a paper in MCP about this last 1 or 2 years ago. She calls it CIRT. I forget what the C stands for. Conserved. Okay, conserved IRT. That's good. So I think yes, and I should say as well, we use a particular kit, these IRT peptides, but there are several kits from other vendors as well. And you can use any of them, really. The only thing we would kind of suggest is that we try to, as a community, stick to the same IRT scale, right? The same IRT dimension that was defined in the first paper that came out a few years back, the first IRT paper, because then it'll just make life easier, right? And the only thing you need to do to establish that is to have your kit spiked in with the other kit and just see where they come on the line, right? Yep. Anything else? No. Oh, one more.
So if you don't use IRT,
yeah.
I was just practicing on a Skyline, not only with the DIA, also the PRM data. Skyline gives you retention time from the library.
Yes.
And that matches your neuro spectra, whether it's close to retention time in the library or not. If you keep the same LC condition constant for
yeah, so the key to that statement is the last one, if you keep the LC conditions the same, right? So if you then decide 2 weeks later that you have more data and you want to use that library on your new data, or a year later, or you change the column, or you want to use that library in a different lab, so if it's within the same data set, then it's probably okay, and it's been all measured together, then you still get a good prediction. But the further you go away in terms of the chromatographic conditions, the more it's worth doing this transformation. Okay, so we better move on because I'm way over time. So next we are going to have the tutorial, but I think first we're going to have like maybe a 5-minute stretching break, and then after that, let's say 10 past, we'll start the tutorial.